Hello and welcome, hopefully welcome back, to the latest in OpenEye Cadence Molecular Science, a series of webinars on computational drug discovery. My name's Paul Hawkins. I'll be your host for today. I am Principal Product Evangelist here at OECMS, and I am delighted to be joined by my colleague Dr. Neha Vithani from our Target Exploration Group. Neha will be presenting today on TargetX, an unobstructed view of pockets. Neha comes to us from Greg Bowman's lab at Washington University, St. Louis, where she worked on cryptic pocket formation and understanding protein allostery through molecular dynamics. So she brings a lot of expertise to the topic. I'm keen to see what she has to say. Before Neha begins, a small note on questions and answers during the presentation. If you have a question, which I hope you will, please pose it through the chat box through the interface. I shall gather the questions at the end and put as many to Neha as we have time available for. If we do not get to your question before the end of the Q&A, we should respond by email as soon as we can. So with that note out of the way, Neha, over to you.
Thank you, Paul, and hello, everyone. Identification of patentable drug molecules is an end goal for pharma business. In this webinar, we will discuss how you can use our target exploration workflow using molecular dynamics and a machine learning model to achieve this goal. To have competitive advantage in finding patentable drug molecules, you might want to work with a disease target that no one else is working on. This requires identification of druggable binding sites in an unexplored disease target. Alternatively, you might want to work with the same disease target that many other companies are working on, but have your own pocket against which you can design your own unique patentable drug molecule. The third option is to work on the same pocket in the same disease target, but identify an alternative pocket conformation against which you can design unique chemical entity that can be patented. So you have three options for getting competitive advantage in finding patentable drug molecules. Regardless of which option you choose, you are starting with a single static structure of your disease target, which is often an apo structure, and you first want to identify all possible pockets in this target. Next, you would want to identify a pocket that has high likelihood of binding to ligand molecules. Finally, you need to select pocket conformations that can yield you high success during virtual screening in separating actives from inactives. At OpenEye, we first developed TargetX method that uses molecular dynamic simulations to identify cryptic as well as non-cryptic pockets. This method achieved 92% success rates in predicting different types of pockets in diverse proteins. Next, we developed TargetX model for predicting ligandability of these pockets to identify pockets that have high likelihood of binding to small molecule ligands. We achieved 67% success rate in enriching known ligand binding pockets among top 3. Finally, we have started working towards identification of conformations that can yield you high success rate during virtual screening in separating actives from inactives. I'll first tell you about the TargetX method for pocket identification. For TargetX method, we start with the apo structure of the protein and determine most collective protein normal modes that describe global dynamics of a protein. Then we drive weighted ensemble MD simulations for enhanced conformational sampling in the presence of Xenon probes in mixed solvent condition. After running the simulations, we identify high occupancy Xenon binding sites for predicting pockets in a disease target. Previously, we have shown that this method was successful in retrospectively sampling the famous cryptic pocket in KRas. This cryptic pocket is known to bind to multiple FDA-approved drug molecules. While this method was successful in predicting this pocket in KRas, just one example is not enough to show utility of TargetX method, because pockets come in all shapes and forms. You can have pockets that pre-exist in the apo structure, And then there are cryptic pockets that transiently open because of protein dynamics. This dynamics can range from sidechain motion to large-scale interdomain dynamics. And in between, you can have loop motion and different types of secondary structure dynamics that lead to pocket formation. The cryptic pocket in KRas is an example of secondary structure motion. Now we wanted to extend the applicability of this method on diverse proteins. So we started by curating ENCORE dataset that consists of pair of apo and holo structures that represent one of the 6 categories of pockets. We computed all heavy atoms of pocket residues RMSD between the apo and holo conformation to quantify differences between the pocket conformation in apo and holo structure. The bar height is representative of crypticity of a pocket and difference in the pocket conformation between the apo and holo structures. For 3 examples, we could not calculate pocket RMSD because a large part of pocket lining residues had missing electron density in the holo structure, implying that these pocket residues are disordered in the holo structure. We call these pockets order to disorder transition types. After curating ENCORE dataset, we started by using TargetX method to see if we can sample pocket formation in these diverse proteins. So we take the apo structure, run normal mode driven weighted ensemble MD simulations, identify high occupancies in on-binding sites, and predict pockets in this diverse dataset of apo-holo structures. We had 92% success rate in sampling the known pockets in the ENCORE dataset. The success was measured based on 2 criteria. First, we compute volume overlap between the predicted pocket by TargetX in the simulated structure and ligand molecule bound in the holo structure. We also calculate the extent to which TargetX was able to sample holo-like conformation. The ENCORE dataset and our validation of TargetX method on this diverse dataset is now available as a preprint, which is also currently under peer review at JAX. So we have developed TargetX method, which can sample different types of pockets with 92% success rate. The next challenge was identifying pockets that have high likelihood of binding to ligand molecules. All pocket prediction methods will give you a large number of pockets in a given target, and ranking the pockets based on ligandability, the likelihood of binding to ligand molecules, is not a new problem. Multiple machine learning models have been developed to rank the pockets by ligandability model. Now, building a reliable and robust machine learning model requires high quality data. All previous machine learning models for ligandability prediction have been developed and trained on biased dataset. When the models are trained on biased dataset, they are bound to give you biased results. To understand what the kind of bias that is present in this dataset, I will show you 2 examples of GRASP and P2 rank model for ligandability prediction. These models were trained on SCPDB and Tran11 dataset. Both these training datasets are heavily biased towards certain types of pockets. SCPDB dataset is dominated by pockets that bind to nucleotides. And Tran11 dataset is heavily biased towards pockets that bind to nucleotides and carbohydrates. These models were not only trained on biased datasets, they were also validated on datasets that show similar bias towards pockets that bind to nucleotides. This results in data leakage between the training and validation set and inflated success rates. Now let's understand what happens when you use these biased models for predicting ligandability of different pockets by the example of KRas. This hollow structure of KRas has 3 different pockets. One of them binds to nucleotides and 2 other pockets bind to drug-like molecules. We remove ligand molecules from this hollow structure and use GRASP model to predict ligandability in these pockets. GRASP model, which was trained on data sets that were biased towards nucleotide binding sites, was able to predict only nucleotide binding site to be ligandable, and it fails to identify the cryptic pockets as ligandable binding sites. The reason for bias present in this dataset is because of how redundancy was removed from this dataset. All these datasets focus on global protein sequence identity or overall 3D structural similarity of the proteins present in the dataset, which does not tell you anything about pocket similarity. We decided to prepare non-redundant dataset by focusing on pocket similarity rather than overall similarity of protein sequence or protein structure. We start by using OpenEye's SiteHopper, which characterizes pockets based on their shape and chemical features. Each pocket is represented by a set of Gaussians that describe their shape and chemical features, and 2 pockets are compared by optimizing overlap between these Gaussians to compute similarity, and the SiteHopper similarity, which measures similarity between 2 pockets, ranges from 0 to 4. 2 pockets with SiteHopper similarity score greater than 2 are considered highly similar. We use a 3-layered approach to prepare a non-redundant dataset of protein-ligand structures. We start with more than 100,000 protein-ligand structures. We first select high-quality structures that have high iridium score at ligand binding site. Next, we apply ligand deduplication to remove pockets that bind to identical ligand molecules in identical position. Finally, we apply SiteHopper similarity-based filtration to remove redundancy by identifying pockets that have highly similar shape and chemical features. The final step of SiteHopper filtration was important for removing redundancy. Without applying SiteHopper similarity filtration, we would have seen certain pockets that are overrepresented in the dataset. So using this approach, we generate more than 1,800 non-redundant protein ligand structures that represent unique and non-redundant pockets. We call this dataset GROOVY dataset. GROOVY dataset was split in training and test set to ensure that there is no data leakage between the two. We take the training data set, and from each protein ligand structure present in the training set, we create a pocket surface patch and a negative control patch. Vertex points present on these patches were featurized based on their geometric and physicochemical features, which also takes their neighborhood and environment into consideration. Next, we train a regression model for predicting ligandability of the surface points using random forest as well as Gaussian process regression method. After training the model, we test it on non-overlapping test dataset. We create a negative control patch and non-ligand binding surface patch from protein ligand structures present in the GROOVY test dataset. TargetX model was able to differentiate between the negative control patch and patches and non-ligand binding patches with 97% AUC. We also split GROOVY dataset in multiple random train and test datasets for cross-validation, and every time we see high differentiation between the predicted ligandability score of negative control and non-ligand binding patches with an average AUC of 97%. We also assign strength of evidence to predicted normalized ligandability score by taking ratio of likelihood that a predicted score belongs to ligandable pocket versus likelihood that a predicted score represents a non-ligandable random surface on the protein. We had taken utmost care in ensuring that there is no data leakage between the training and the validation set. We wanted to take one more step to ensure that the TargetX model has not simply memorized the training set. If the model has memorized the training data set, we would expect high predicted pocket score for the surface patches that look very similar to the examples present in the training set. However, we don't see any strong correlation between the predicted pocket score and its similarity with the examples present in the training set, suggesting that the TargetX model has not simply memorized the training set, but it has learned something useful from the training data. How the TargetX model was built on the GROOVY dataset is discussed in the preprint that is available online, and it is also currently under peer review. Now that we have built TargetX model, which can predict ligandability and rank the pockets based on the likelihood of binding to ligand molecules, we wanted to use it for ranking the pockets that were predicted in our ENCORE dataset using TargetX method. So we take all the pockets that are predicted by TargetX method for each example in the ENCORE dataset and rank them based on ligandability. We achieve 67% success rate in ranking known ligand binding pockets among top 3. So we have TargetX method, which can predict pockets, TargetX model, which can rank the pockets and help you identify good pockets that have high likelihood of binding to ligand molecules. Can we use it for exploration of new targets? To answer this question, we started with retrospective prediction of pocket in NKG2D, which is relatively unexplored disease target. This protein was known to be clinically relevant for autoimmune disease and inflammation. However, it has not been explored for identifying small molecule ligands. Here I'm showing you relatively flat surface of the apostructure of specific region in this protein NKG2D. Recently, an extensive experimental study conducted at Janssen identified a cryptic pocket that would transiently open because of protein dynamics. This pocket was identified by running extensive screening studies, and it was detected in experiments only when the right molecule could bind to this cryptic pocket. It was found to be particularly challenging to identify this pocket by computational studies like induced fit docking and 50 microsecond long brute force molecular dynamic simulations. We wanted to see if TargetX method can retrospectively predict this pocket if we start from the apostructure. And within 4 microseconds of weighted ensemble MD simulations, we start seeing pocket opening. And TargetX not only samples the pocket opening, it also ranks it by ligandability. This example of cryptic pocket in NKG2D is also that of secondary structure motion among our ENCORE dataset. So now we have TargetX method, which can help you identify good ligandable pockets in unexplored disease targets. The next challenge is identifying conformations that can yield you high success during virtual screening. In our ENCORE dataset, we had different examples that show high variation between the apo and holo pocket conformation. When we use weighted ensemble molecular dynamic simulations, we sample pocket conformations that look more holo-like compared to the starting structure. Can we take advantage of the conformational heterogeneity of pockets that is sampled by running TargetX method? To answer this question, we wanted to select an example where we have been able to achieve this huge conformational heterogeneity by running weighted ensemble MD simulations, and we have knowledge of large number of active molecules. So to conduct this experiment, we selected the example of Bt kinase, where we have sampled diverse pocket conformations and we have availability of multiple known active molecules. So we select 370 known active compounds and generate 9,000 pattern-matched decoys on DIRT-Z. We select apopocket conformation as well as hollow pocket conformation and use these and dock these compounds against these two pocket conformations. We find that hollow conformation was able to separate actives from inactives with much higher success than the apopocket conformation. When you run TargetX, we also give you a pocket representative structure. In case of Bt kinase, this pocket representative structure looked a lot like apopocket conformation. Therefore, it had lower success in separating actives from inactives. We decided to select few more conformations that look more hollow-like as sampled by weighted ensemble molecular dynamics, and we run the same docking experiment and see improvement in AUC when we run docking against these pocket conformations. So this retrospective analysis shows that conformational heterogeneity generated by weighted ensemble MD simulation can be advantageous in improving success rate during virtual screening. We are now exploring different ways in which we can prospectively identify these conformations that can be successful in separating actives from inactives. To summarize, we have developed TargetX method, which uses weighted ensemble MD simulations in presence of Xenon probes to identify all kinds of pockets, and we achieved 92% success rate when it was applied to diverse data set present in ENCORE dataset. Finally, we have TargetX model, which can rank these pockets based on their likelihood of binding to ligand molecules, which gives us 67% success rate in enriching known pockets among top 3. And now we are working towards identification of pocket conformations that can be useful for separating actives from inactives during virtual screening. Finally, I would like to thank everyone who made these projects successful. I would like to thank scientific leadership at OpenEye for guiding the projects in right direction. These projects would not have been possible without scientific and technical contributions from the target exploration group at OpenEye. I would also like to thank scientists from 3 different pharma companies who gave us challenging test cases on which we can apply TargetX method. And as a reminder, the work that I discussed today is available as preprints that you can read for more details. Thank you, everyone, and I'm ready to take more questions.
So Neha, thanks for that. Fascinating as always. A couple of questions have come in. I'll warm you up with an easy one. Why did you pick Xenon as your probe to be a part of that mixed solvent simulation?
Usually, organic molecules are used, or organic fragments are used as probes, but we decided to go with Xenon, which is a single atom. It can have rapid diffusion. It can quickly bind to diverse pockets. There was also an experimental study conducted in 1980s on where X-ray crystallographic structures were determined in presence of Xenon, which showed that Xenon is capable of binding to diverse pockets, including those that are lined by hydrophilic residues. It is also a general anesthetic, and it is known to bind to different proteins to act as general anesthetics. So we had evidence to know that Xenon can bind to diverse pockets, and it can have rapid diffusion. So that was the reason for us to use Xenon.
Seems ideal for a molecular dynamics simulation probe. Cool. Okay, a slightly sort of broader question. You sort of talked about the TargetX sampling approach to generate candidate pockets, and then the TargetX model to try to assess which ones are most likely to be ligandable. You talked about them as a pair. Is it possible to use the TargetX ligandability model standalone to look at pockets that perhaps don't come from molecular dynamic simulation or don't come from TargetX?
Yes, it can absolutely used for identifying pockets on a single static structure. The way you can use it is by taking the structure, predicting ligandability of vertex points on the entire surface of the protein, and then identifying clusters of ligandable surface points, which will point you to the pockets that are likely to bind to ligand molecules.
So provided you have coordinates for the protein atoms, you can use the TargetX model no matter where those coordinates came from. That's cool. Slightly more in-depth question, so I'll give you some time to answer this one. How did you come up with the particular progress coordinates that you used, and what made you choose weighted ensemble simulation as your simulation method of choice? So that's actually two questions I sort of crammed into one because I think the two of them are related. So firstly, progress coordinate choice, and secondly, why weighted ensemble specifically?
I'm going to answer the question in reverse order. So one can use plain molecular dynamic simulations without much success in sampling different types of diverse large-scale motion. That's where we would want to use enhanced sampling method. There are different enhanced sampling techniques available, but we decided to use weighted ensemble MD simulations because they do not perturb underlying Hamiltonian. So without adding any bias to the potential, we can sample diverse conformations. Now, the challenge is which progress coordinate will you use for enhancing the sampling using this technique. When you don't know where the pocket is present, it's very difficult to decide on appropriate progress coordinate. Now, because pocket can be present anywhere, we wanted to enhance sampling of entire protein. So we select normal modes that will maximize sampling of entire protein. And that's where we decide to use most collective normal modes that describe global dynamics of the protein rather than focusing on a specific region or taking any information into consideration. Now we can globally search all possible pockets for a disease target.
So you're attempting to use both of those to minimize or to eliminate bias in the calculation and allow the protein to explore its landscape as fully as possible.
That's right. No bias, no prior information required.
Neha, thank you very much for that. We're about at the end of time for today's presentation, so thank you again for that. Thank you all very much for attending today's webinar. I hope you got as much out of it as I did. I look forward very much to seeing you at the next of our webinars here from OpenEye Cadence Molecular Sciences. So until then, goodbye.